In this video we will discuss about a new paper about this pancreatic cancer, where they have shown that this natural killer cell, which is one of the immune cells, are present in the tumor microenvironment of the pancreatic cancer cells, and they are active. So we will discuss in this video about their abstract, and in the subsequent video we will discuss the results that comes to this conclusion. So this is the first figure on the top left hand side. Shows that this tumor microenvironment usually has this high cancer associated fibroblast and collagen content that will block NK cell accumulations, and these are the NK cells. This yellow dot, it blocks the NK cells accumulations around the tumor microenvironment. Basically, this high cancer associated fibroblast and collagen content act as a physical barrier that helps in the immune evasions of the tumor that are present within the tumor microenvironment. And if this CAF and collagen contents are reduced, then it will permit the NK cells accumulation. When the physical barrier is not strong, then NK cells can permit and interact with the tumor cells and trigger the immune response. And the reasons, in the first case, the reasons why the NK cells is not been able to interact with the tumor cells is that the CD44 receptors that are usually present on the NK cells binds and interact with the collagen that is present in the cellular matrix, thereby preventing them from interacting with the tumor cells, pancreatic tumor cells. And as long as the NK cells does not interact with the tumor cells directly, it will not be able to trigger immune response. However, the researchers, what they did is that they neutralized this CD44 on the NK cells, and then check the invasions, or check this CD44's accumulations in the tumor microenvironment. They are able to see that when the CD44 is neutralized, it will promote the NK cells invasion in the tumor microenvironment, and it can interact with the pancreatic tumor cells and then trigger the apoptosis. And these things, in order to do these things, they took out around human patient samples to analyze this phenomena, that is the natural killer cells presence in the tumor microenvironment. And here the representation is in the way that the tumor cells that are represented by the circles, sorry about that, the tumor cells are represented within these circles that are not shown here, but it is circulated by this we can say this epithelial duct. And PDAC, that is pancreatic ductal adenocarcinoma, is one of the most commonly found pancreatic cancers in the patients. And this yellow this tumor microenvironment parts that is represented by this yellow color is the fibroblast, and it also has this anti-CD44 that is used to neutralize this NK cells, that is shown here, the anti-CD44s. It can neutralize the NK cells or natural killer cells. Along with that, it also shows that this ECM is there, that is extracellular matrix. And during this pancreatic tumor cells or pancreatic ductal adenocarcinoma, what happens is that a high dense cancer associated fibroblast and collagen contents are formed that act as a physical barrier, and thus results in the immune evasions from the immune cells that will not be able to pass this physical barrier in most of the cases. That is the reasons tumor microenvironment That is the reason these pancreatic tumor cells are considered as more aggressive of all kinds, or one of the most aggressive pancreatic cancers. So we will discuss the results, how they come up to this conclusion that if the CD44 is being neutralized in the NK cells, and it can promote NK cells invasions, or NK cells can penetrate through the tumor microenvironment. If you want to understand more about this paper in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.